Welcome to the introduction to Monera, or our bacteria, which is our first true kingdom that we are going into. Now, just as a review, bacteria are prokaryotes or prokaryotic, and this means that they don't have organelles that are surrounding, or they don't have membranes surrounding their organelles, and they also don't have a nucleus. And you remember from the chart that we did on the first day of this unit that they're split into two kingdoms, the eubacteria, which are referred to as the true bacteria, and the archaebacteria, which can be thought of as ancient bacteria. So here are some eubacteria, and then right here is an example of a hot spring where you might find archaebacteria. Here's just a diagram of some of the diversity we can find in these two groups. So here's the archaebacteria and the eubacteria. You'll notice that there's these gram-negative and gram-positive. By the end of this presentation, you should know the difference between the two, and then you'll also be able to determine how we name and describe these different shapes and arrangements of bacterial cells. So bacteria parts, most of them you will be familiar with. The cell wall you'll be familiar with from plant cells, but in bacteria, some of them are rigid and others will be flexible. The cell membrane is the same as other cells that you've learned about. Cytoplasm is also the same as other cells that you've learned about. DNA, in bacteria, they have single circular chromosomes, and that's located in the cytoplasm. Remember, they don't have a nucleus. They also, in addition, so that's kind of this big red glob, and then they also, in this little blob, have plasmids, which you'll remember from the applied genetics unit. And then a capsule, which is only on some bacterial cells. That's the red in this diagram, and that's a thick gel-like protective coating. Pili, which are short hair-like protein structures on the surface of some bacteria, which is the green in here, and that helps them stick to host cells. And then flagella, which, remember, some bacteria can move. We talked about that in the kingdoms chart. And this is long protein structures so that's the flagella right there that will propel some bacterial cells. So now I'd like you to pause the video and I'd like you to draw your own version of this bacteria and label the parts on your notes packet. There's a box for you to draw that in. Now that you've returned, we'll go into the two different groups a little more specifically. So the archaebacteria, these guys are really thought to be relatives of some of the earliest life forms. And they're a pretty small category. We can split them into three main groups. The methanogens, this group, for this group, the oxygen is poison, so they must live in anaerobic conditions. And they convert hydrogen and carbon dioxide into methane gas for energy. And then the extreme halophiles are considered salt-loving bacteria, so they'll live in environments with very high salt concentrations, and these concentrations would kill most bacteria. They use that salt to then generate ATP for energy. And then the thermoacidophiles, you can probably tell from the name that they live in areas that are extremely acidic and then also have extremely high temperatures. So this can be up to 110 degrees Celsius. So those are our archaebacteria. Our eubacteria, these are the ones that we commonly think about when we think of germs. So there are definitely eubacteria that do very positive things for us, and that'll be familiar to you with yogurt, for example. So these guys are the most of the world bacteria. They kind of bring about most of the bacterial diversity. They, depending on who you talk to, are divided into 12 phyla. So I said 12-ish phyla. They are most widely distributed organisms that we know of, though they do generally thrive in temperate environments. The normal human body temperature is a great example of a temperate environment for these bacteria. And E. coli, or Escherichia coli, which is the bacteria in the background, that's a rod-shaped bacteria, and that's common in our digestive systems. There are different strains, and some are good bacteria, and some, for humans, are not so good bacteria. Now, we classify eubacteria in several different ways. So the first one is by shape. I'm going to take that away for a minute. So notice in this background image we have different shapes of bacteria, and then we have different, within those shapes, kind of different arrangements. So there are rod-shaped bacteria, round bacteria, and then kind of spirally bacteria. So for shapes we have the cocci, which are round, bacilli, which are rod-shaped, and sporili, which are spiral-shaped. Now we can also combine those with different prefixes that describe the clustering. So diplo bacteria have two bacterial cells together. Staphylo have a cluster of bacteria, and sporili are, that should say strepto, and that does say strepto in your notes. So these are indicate filaments, so bacteria that are kind of back to back, like in this image. So based on the shape and clustering names that you just learned, what does Streptococcus look like? So pause for a minute and draw that in the empty box on your notes sheet. Now we can also classify them based on their respiration. We have the obligate anaerobes, and these guys cannot survive in the presence of atmospheric oxygen. So anaerobes, remember, is without oxygen. Obligate, meaning they must have no oxygen. Our facultative anaerobes can live with or without atmospheric oxygen. So facultative is like, well, they'll take advantage of no oxygen or oxygen, but they can survive in either. And then on the opposite side, we have our obligates, so they require aerobes, they require oxygen. And these guys can't survive without atmospheric oxygen. Now, another way that we can differentiate between our bacteria, and we'll do in class on Friday, we'll do an activity for this, is gram staining. So what color the bacteria stain if you use two different types of stains. So we can split bacteria into two different groups in this way. One of them has a thick peptide layer, and one of them has a thin layer. And those bacteria will stain differently and hold on to different stains, which can differentiate between the two. So again, two types of cell walls in the eubacteria. The gram-positive bacteria have a thick peptide layer, and that helps them retain this crystal violet color that you see, the bluish, and they'll appear purple when viewed under a microscope. So if this is our plasma membrane, then this is our thick peptide layer. Then we have gram-negative bacteria, which have a thin peptide layer, and a space surrounding it, so the cell wall is actually more protected. So it's maybe the opposite of what you would think. And they take up the pink stain, and it will appear pink when viewed under a microscope. So here's some pink gram-negative bacteria. For the gram staining, and again we'll do this in class doing a virtual example of this in class. You will first put cells on a microscope slide that are stained with purple dye solution called crystal violet, which is happening in this step 1. Then you wash the cells with water. Then you add an iodine solution. Then that slide is washed with alcohol. And then finally you'll restain the cells with a pink dye solution called safranin. So the gram-positive bacteria will stain violet after this, and then the gram-negative bacteria will stain purple pink or red. And let's watch this animation to kind of see how that's done.
The gram stain is the most widely employed staining method in microbiology. It is a differential stain because it divides bacteria into two classes, gram-positive and gram-negative. In the first step of the gram stain procedure, cells from a fresh culture are transferred to a clean slide and allowed to dry. If the cells are on an agar plate, they should first be transferred to a liquid medium for dilution. The cells should form a thin, barely visible film. This can be achieved by smearing cells obtained from the surface of an agar medium or from a liquid culture. Fresh cultures must be used because as cells age, they lose their ability to retain the stain. The cells are then fixed to the slide by passing slightly above the flame of a Bunsen burner. After passing above the flame, the slide should feel warm when touched to the back of the hand, but should not be too hot. The fixed cells are then stained with the basic dye, crystal violet, for 30 to 40 seconds. The slide is then rinsed with water to remove excess stain. At this point, all cells appear purple under the microscope. Next, a solution of Graham's iodine is added and retained on the slide for about 1 minute. The iodine combines with the crystal violet to form a di-iodine complex, thereby decreasing its solubility within the cell. At this point, the cells still appear purple. The cells are then decolorized by washing with ethanol or acetone. This is the differential step. Gram-positive bacteria retain the crystal violet, whereas gram-negative bacteria do not. The ethanol or acetone should be added drop-wise, with the slide tilted at an angle until the drop coming off the edge of the slide just starts to become colorless. Even gram-positive cells can lose the crystal violet iodine complex during prolonged excessive decoloration. Excess ethanol is then washed off with water. When viewed under the microscope, gram-positive cells appear purple and gram-negative cells are colorless. Finally, the rinsed cells are covered with the counterstain safranin for 20 to 30 seconds. This stains the gram-negative bacteria pink. After rinsing with water, the slide is dried with filter paper. When viewed microscopically, the gram-positive bacteria are purple and the gram-negative bacteria are pink. Generally, the gram stain correlates with the cell wall structure among the bacteria. The ethanol is thought to shrink the thick peptidoglycan in gram-positive cells, thus retaining the dye. The thick, dehydrated peptidoglycan layer of gram-positive bacteria appears to be a permeability barrier, preventing the loss of the crystal violet iodine complex. In contrast, the peptidoglycan in gram-negative bacteria is very thin and has large pores. Ethanol may extract lipids and increase the porosity, thus removing the crystal violet iodine complex.
Okay, so that was a little more in depth, but in class you'll look at, you'll actually get to investigate whether there are positive bacteria that you want or bacteria that you don't want in a batch of yogurt. Now here's an image of the end result. So we have our E. coli, which is in the first picture, and that's gram-negative. And then these last two are gram-positive, so there's Staphylococcus and Bacillus cereus, which are both gram-positive bacteria. And they appear with the violet color. And we are actually going to stop there today, so I'm cutting the film a little bit short for you. What I would like you to do for your summary in this notes packet is to summarize how. Actually, I'd like you to just summarize, kind of do a general professional paragraph summary, and I'll see you on Friday.